Good morning, afternoon, or evening, depending on where you're located. I would like to start by thanking the organizers for inviting me to be here today to speak with you about our research over the past 8 years, defining the effects of space radiation on the hematopoietic and GI systems. In deep space, astronauts will be exposed to a unique radiation environment consisting of solar energetic particles, or SEP, which are intermittent release of highly energetic particles, mostly protons, from the Sun. These particles travel at near the speed of light and arrive at Earth within only minutes of the event on the Sun. Even more problematic are galactic cosmic rays, which are comprised of high-energy particles, aka HCE, which are mainly high-energy protons, and atomic nuclei, or atoms stripped of their electron shell. GCR originates outside the solar system, likely from supernovae and active galactic nuclei. In contrast to SEP, GCR radiation is continuous and omnidirectional. To give you an idea of how serious this problem is, NASA currently estimates that every nucleus in every cell of an astronaut's body will be traversed by a proton once every 3 to 4 days, and by a high LET ion once every 3 to 4 months while beyond low Earth orbit. Unfortunately, we don't really know much about the biological effects of SEP and GCR radiation, and the reason for this is that on Earth we are actually almost completely protected from SEP and GCR radiation as a result of the atmosphere and the magnetic field or magnetosphere that surrounds the Earth and acts as a shield. Now, with all the different tissues in the body, why would we choose to focus on only the hematopoietic and GI systems? Both systems are very radiosensitive, and damage can be life-threatening in a very short period of time. Leukemias are one of the most frequent radiogenic cancers, and colorectal cancer can also be induced by ionizing radiation, or IR. Importantly, leukemias have a short latency. As such, they could arise and progress sufficiently to compromise a mission. To begin defining the effect of space radiation on the human hematopoietic system, we collected bone marrow from healthy astronaut-age human donors and isolated hematopoietic stem cells, or HSC, by magnetic sorting. These HSC were then transported to the NASA Space Radiation Lab, NSRL, where they were exposed to protons, iron ions, and sequential proton plus iron ions. Aliquots of these HSC were also exposed to gamma rays as a reference, or left unirradiated. We opted to include sequential proton and iron ion exposure because of prior work from Betsy Sutherland that had shown marked enhancement of DNA damage when human cells received sequential exposure with protons followed by iron ions, but interestingly not vice versa. Greg Nelson's group also showed similar outcomes in the hippocampus of mice irradiated in vivo. This is important because this is actually what the astronauts would see based on the fluence of these ions in deep space, i.e. each one of the nuclei would be hit by several protons before it would actually encounter an iron ion. Upon return from NSRL, the colony-forming potential of the human HSC were assayed using the luminescence-based halo system. As you can see here, exposure to SEP or GCR radiation markedly reduced the formation of the most primitive HPP-CFC and CFU-GEM colonies. Interestingly, sequential SEP plus GCR exposure led to an at least additive or perhaps synergistic reduction in the output of these colonies. While colony-forming assays are a mainstay of hematopoiesis research, the gold standard for assessing HSC function is to test their ability to repopulate immunodeficient mice. We therefore repopulated NSG mice with human HSC exposed to each of the doses and ions of radiation and followed these mice long-term, measuring the presence and levels of human cell engraftment as well as the differentiation and lineage commitment. As you can see here, exposure to protons, iron ions, and sequential proton plus iron led to marked skewing of the in vivo differentiative potential of the human HSC, with an expansion of the lymphoid compartment at the expense of the erythroid lineage. The most remarkable finding was the presence of pronounced splenomegaly in the mice repopulated with human HSC exposed to iron ions, with some spleens being roughly 30 times the normal size. Flow cytometric analysis confirmed that the enlargement of the spleen was due to population with human cells. Histopathologic examination of the enlarged spleens revealed the presence of human T-ALL, and I would like to draw your attention to the fact that this is the first and the only human cancer ever reported as a result of exposure to GCR radiation. Whole exome sequencing of the human HSC exposed to each radiation type identified numerous mutations specific to each radiation type and demonstrated that these alterations were similar between HSC from two different human donors. We also found that SEP and GCR radiation appear to have a predilection for introducing mutations in genes that have been implicated in hematological malignancies. For those who would like more details, we published these findings. We also published an additional paper showing that SEP-GCR radiation can induce biological bystander effects on human hematopoiesis by altering the cells of the marrow microenvironment. While these studies were an important first step to understand the risks to the human hematopoietic system as a result of exposure to SEP-GCR radiation, because we had irradiated the cells in vitro, we worried that some of the effects might be different if radiation occurred in vivo. To address this issue, we began studies in which we first repopulated NSG mice with normal healthy HSC from human donors of typical astronaut age, waited for the engraftment to stabilize, and then brought the mice harboring a human hematopoietic system to NSRL to expose them to whole-body SEP-GCR radiation. Shown here are dot plots of the isolated HSC population from a typical human donor on the top, and the engraftment of human cells within the hematopoietic system of these mice. Over the course of our studies, human engraftment has averaged about 16%. Over the past few years, we have humanized 495 mouse avatars with HSC from 8 different human donors, and have brought these mice to NSRL to expose them to protons, a variety of HDE ions, and the newly created GCR simulator, and the details of these exposures are shown in this slide. Analysis of the bone marrow of these humanized mice at 4 to 6 months post-irradiation demonstrated a similar skewing of lineage commitment to what we had seen when the HSC were exposed in vitro prior to repopulating mice, namely a marked expansion of the lymphoid compartment at the expense of the erythroid and myeloid lineages. This effect was most pronounced with heavier ions such as oxygen and iron. Within the spleen, exposure to protons or GCR ions led to a pronounced shift in the T cells to a pro-inflammatory CD8 positive phenotype. As we had seen in our prior studies, humanized mice exposed to GCR ions in vivo also exhibited pronounced splenomegaly. Staining with human-specific Ku80 antibody confirmed that the abnormal nodules within the enlarged spleens were human. As shown in the bottom panel, we then developed a method of sorting human hematopoietic cells back out of these mice, making it possible for us to now perform genomic and transcriptomic studies on the human hematopoietic cells within these mice to define the changes that have occurred as a result of SEP-GCR exposure and are driving this pathological process. As you can see in this slide, the incidence and severity of splenomegaly was a direct function of the radiation type, with the heaviest ions, i.e. oxygen and iron, inducing the highest frequency and the most severe splenomegaly. Moving forward, the slides from the enlarged spleens have been viewed by clinical hemopathologists at Wake Baptist and have deemed to be very likely malignant. Licensed veterinary pathologists are currently reading the spleen sides and slides from other tissues of these mice to diagnose the nature of the malignancies present in the humanized mice exposed to each radiation type and dose. Thus far, they have confirmed both leukemias and lymphomas, depending upon the ion to which the humanized mouse was exposed. So to summarize what we found in the hematopoietic system to date, human hematopoiesis is markedly altered following in vivo exposure of human HSC to Mars mission-relevant doses of protons, or SEP, oxygen ions, iron ions, and the 5-ion simulated simplified GCR simulator. GCR exposure in vivo appears to induce leukemia or lymphoma, as was seen when HSC were exposed in vitro and then transplanted. This has now been seen with 4 different human donors. Analysis of humanized mice that were exposed to silicon ions are currently ongoing to assess whether there's a possible LET effect. Prior to moving to the GI system, I would also like to draw your attention to other ongoing studies in our lab in which we developed a human bone marrow on a chip to enable us to study normal and malignant human hematopoiesis and define specific HSC niche interactions. This system could provide a much-needed alternative to studying human HSC within a xenogenic mouse environment. To study the effects on the GI tract, we performed studies in which wild-type mice were exposed to the same doses and species of SEP-GCR radiation as the humanized mouse avatars. We then collected the small intestine at various time points post-IR and performed a battery of assays to define the alterations that occurred as a result of exposure to space radiation. As you can see in these images, exposure to SEP protons, oxygen, or iron ions in vivo led to massive lymphocytic infiltration and swelling of the lymph nodes within the small intestine within 24 hours of exposure. These exposures also caused vascular congestion and bleeding within the GI tract, which you can see here circled in red. Staining for the tight junction protein, Claudin-3, demonstrated that exposure to protons and especially iron ions led to marked disruption of the epithelial barrier within the small intestine. A custom script was then used to quantify these changes, and the results are shown in the graph in the middle. Staining for PCNA and Musashi-1, a marker for intestinal stem cells, showed that IR exposure triggered proliferation within the crypts, and also showed that iron ions perturbed the ISC pool, which can be seen in the bottom right. In an effort to identify pathways that could be targeted to prevent the damage we had observed in the hematopoietic and GI systems, we performed transcriptomic analysis on human cells exposed to gamma rays, protons, and iron ions, and identified differentially expressed genes using each donor's unirradiated cells as an internal control. These analyses identified multiple pathways that are all affected by the compound curcumin, which is found in the spice turmeric. Treating human HSC with curcumin, either prior to, in the middle panel, or even following, in the right panel, exposure to IR led to marked radioprotection or radiomitigation, restoring much of the lost colony-forming potential of these cells. Unfortunately, curcumin has very poor water solubility and bioavailability in vivo. We therefore worked with Matt Coleman and Paul Wilson at UC Davis, who developed nanolipoprotein particles, or NLPs, that they showed could be efficiently loaded with curcumin, making it water-soluble and allowing the agent to be administered in vivo. Quite remarkably, pre-treating the humanized mice with the curcumin-loaded NLPs 18 hours prior to exposure to SEP or GCR radiation significantly reduced both the incidence and the severity of splenomegaly, suggesting radioprotection to the hematopoietic system. The effects in the GI system were even more pronounced, with CNLP pretreatment reducing the number of apoptotic cells, shown on the left, and almost completely eliminating the induction of double-strand breaks following exposure to gamma rays, protons, or iron ions, shown in the right panels. So to summarize our findings in the GI system, we've shown that exposure of the native murine GI tract to Mars mission equivalent doses of protons or SEP, oxygen ions, iron ions, and the 5-ion simplified GCR simulator in vivo leads to marked alterations in the small intestine, including induction of double-strand breaks, apoptosis, disruption of barrier function, bleeding, and lymphatic swelling. Licensed veterinary pathologists are currently analyzing all sections from small intestine to provide a clinical diagnosis of the effects of each radiation type on the GI tract. Looking at the impact of our studies for space travel, it's our hope that the successful completion of our studies will greatly enhance our understanding of the effects of Mars mission relevant doses and fluences of space radiation on the human hematopoietic and GI systems, thereby facilitating NASA's risk assessment. It will define the molecular mechanisms responsible for the observed effects and identify biomarkers that could then be used to assess astronaut exposure and resultant damage. And we're hoping to finalize a stable lyophilized form of an orally administered countermeasure that could protect against the harmful effects of space radiation, thereby enabling safe, long-duration missions in deep space. Importantly, our findings are not only of importance for astronauts in space travel, they also have an impact for those of us living on Earth. A better understanding of the dose and the LET effects of ionizing radiation and the mechanism by which high LET ions damage DNA would enable safer and more effective radiotherapy for cancer patients on Earth. The countermeasure that we're developing could potentially be used to mitigate the off-target toxicities that occur in the hematopoietic and GI systems of patients who receive radiotherapy. And similarly, the biomarkers that we identify could be used to screen civilians following accidental exposure. I would like to finish by acknowledging all of the people who have contributed to this work. My partner in the lab and in life, Dr. Graça Almeida Porada, all the members of our lab group and our collaborators at the Wake Forest School of Medicine, as well as Paul Wilson and Matt Coleman and their teams at UC Davis, and Peter Gaida and Adam Russick at NSRL. And of course, none of these studies would have been possible without the support of NASA's Space Biology Program, the Human Research Program, and NASA's Translational Research Institute for Space Health. Thank you very much for your attention.